Hi everybody, this is Dr. Kat again from Central New Mexico Community College. We're going to spend one last video on this diagram that you've seen over and over again that relates to the neuromuscular junction. And we're going to focus this time on how ion concentrations are restored. So the restoration of ion concentrations is the topic of this video. By now you know, as we move back over here, that when an action potential arrives from the central nervous system at the level of our bulb here that belongs to an axonal terminal, we're going to see that calcium will follow its concentration and enter our axonal terminal, or the bulb of the axonal terminal. And that triggers the release of acetylcholine from the synaptic vesicles via exocytosis. Now, how does that calcium enter into the axonal terminal? Via voltage-gated ion channels, because when this action potential arrives, it brings with it a severe change in the potential, a severe strength of depolarization actually, and that triggers the opening of these calcium voltage-gated ion channels. So we've seen sodium and potassium voltage-gated ion channels on the muscle cells membrane, better called the sarcolemma, but we see on the cell membrane of our axon's terminal here also calcium voltage-gated ion channels. We have primarily chemically gated ion channels at the level of the motor end plate, because that's where our chemical binds, namely our neurotransmitter called acetylcholine. We may have some voltage-gated ion channels mixed in here. That is our sodium and potassium voltage-gated ion channels that are very numerous along the length of the rest of the sarcolemma, more away from the motor end plate, down into the T-tubules. And then finally, we also have calcium voltage-gated ion channels in the membranes of our terminal cisternae. Remember, when this action potential travels down into these T-tubules with its strong strength of depolarization, that will yet again trigger the opening of voltage-gated ion channels, calcium voltage-gated ion channels again, as well as in this terminal cisterna. So we see calcium voltage-gated channels in the membrane of our axonal terminals bulb. We see calcium voltage-gated channels in the membranes of our terminal cisternae. We see primarily sodium and potassium voltage-gated ion channels down the T-tubules and along the length of the sarcolemma, not so much probably within the motor end plate. Okay, so when all of these ions are moving along their concentration gradients through these ion channels. So think of these ion channels just as little doors that will ion channels with little doors that will open up, and then our ions just follow their gradient. Wherever there's more of an ion, the ion will move away from the area where there's more of it to an area where there is less of it. But once this is all needing to stop, as in once there is no more trigger anymore from the central nervous system, once there's no more action potential coming down from the central nervous system, and therefore no more acetylcholine is released, all of these ion concentrations are messed up, and that's where ion pumps play a role. So all my circles represent ion pumps. Now we have various ion pumps, as I've mentioned before, but what we need primarily at the level of the axonal terminal is a calcium ion pump. All of this calcium that moved into our bulb here now needs to be pumped back out. Similar principle here with our sodium potassium. Our sodium rushed in through the voltage-gated ion channels, so the pump must pump the sodium out. And the same pump, the sodium potassium pump, will pump the potassium back in. Now these pumps are always busy, by the way. They're always going. Same with the leakage channels I've mentioned, and I haven't illustrated those here, but you should pretend that there are some channels who have their gates open all the time. And so ions can just sort of move back and forth along those along their concentration gradients, depending on what their gradients are, through those leakage channels as well. So it gets very complicated. Not only that, all cells have sodium-potassium pumps in their cell membrane. So why maybe we see primarily here at the level of the axonal terminal calcium pumps, we probably also see sodium-potassium pumps. So the pumps are always going. But when there is no more movement through these chemically gated ion channels and voltage-gated ion channels, we're going to see that the pumps are eventually going to have an opportunity to catch up with restoring the ion concentration gradients. I'm going to mention one last thing, even though the focus of this discussion was to be on the restoration of ion concentrations. I'd like to also address what needs to happen here at the level of the synaptic cleft when there is no more action potential coming down. So if this signal is not arriving anymore, clearly our voltage-gated calcium ion channels will not open up anymore, no more calcium flowing in, no synaptic vesicles merging and releasing the neurotransmitter called acetylcholine by means of exocytosis, etc. So we don't have any movement of ions through these ion channels anymore. But the acetylcholine might still be floating around here. It's not going to just suddenly disappear. And that's why we have present inside or floating around or nearby the motor end plate, I should say, a very special enzyme that is called acetylcholine esterase that is always there. It is always there. And it's always going to try to break down acetylcholine. So acetylcholine will bind to its receptors, but pretty soon acetylcholine esterase will take it apart into acetyl and into acetyl and into choline. And your book will show you how some of those products will return to the axonal terminal for recycling and etc. We won't spend any more time on that. But this enzyme plays a very crucial role in removing the acetylcholine from the synaptic cleft. So when there's no more action potential coming through and therefore less and less acetylcholine is being released, or none actually, eventually that acetylcholine esterase molecule or enzyme has the opportunity to destroy all of the acetylcholine. It works really fast, and you'll see that this is an important thing to know down the road when we get to the nervous system. So acetylcholine esterase is an enzyme that's always working. Our ion pumps are always working. Doesn't matter which ion pumps. Are chemically gated, and our voltage-gated ion channels, on the other hand, they only open when they receive the right stimulus. Chemically gated ion channels need a chemical stimulus. Voltage-gated ion channels need a voltage stimulus. None of these ion channels, except for the pumps, need ATP. All of these ion channels, that is the leakage ion channels, your chemically gated ion channels, your voltage-gated ion channels, they only work by allowing ions to flow along their concentration gradients. Ion pumps will always use ATP to move ions against their concentration gradient, something very important to remember. So on that note, thank you for watching.